Good morning, everyone. I welcome next webinar of extractable and leachable, which is very interesting topic. Allow me to introduce myself. Myself, Milan Modi, and I'm working as a director of operation at General Pharma Services. I'm having more than 15 years of experience in the field of clinical research. And here, me along with my colleague, Mr. Ketal Prajapati, are going to discuss very important and very interesting topic that is extractable and the leachable. So we are going to split this topic in a two part. First part includes the introduction, understanding of the extractable and the leachable, and the later part, which is going to be a second webinar that covers the method development, validation, and the interpretation. So first topic we will start with the introduction and the understanding. I request my colleague Mr. Ketal Prajapati to introduce himself and start the discussion.
Thank you, Milan, and very good morning to all of you, and warm welcome to all of you. My name is Ketal Prajapati, and I am working as an associate director for the GXP services at Zinoware. I am having more than 20 years of experience in the field of preclinical, clinical, and analytical support. So let's start our discussion on extractable and leachables. So today we will discuss more on how to identify extractable and leachables, and after identifying how to interpret the data that we will cover in the next webinar. Thank you, Milan.
So let's start the discussion with a very basic question. That is, what is the purpose of having the extractable and the leachable studies, and what would be the objective? Why it is required? If you can focus on very basic question first.
Thank you, Milan. The objective is to ensure the product safety in the pharmaceutical packaging. So nowadays we are hearing that many products they were taken back from the market after post-market approvals. And even for this product, during the method development of this product, all the toxicity data were generated and there were no impurities found which is toxic. So what was the reason and what is the consequences that lead to the this type of results? So after investigation for the many products, it was found that the impurities which are toxic and found uh, after post approval is because of the container closure system that interact with the drug molecule. So that's why it is very important to evaluate the acceptable leachable before marketing of a drug.
So basically, not only the pharmaceutical product which contribute to the impurity and the safety of the drug, but also container closure system which can also impact on the safety and the toxicity, right? So this is very interesting. Let's discuss further.
So let's start with the introduction about the acceptable and leachable. So what is acceptable and leachable? So when we are talking about the acceptable, the compounds that can be extracted from the packaging or a process components using a aggressive conditions. It means we have to give extra rubber condition during the extraction for the evolution of the extractables. Whereas the leachable, generally we have to mimic the conditions such as drug life cycle. So compounds that are migrate into the drug product under the normal condition over the time can be identified as a leachable. So the important part is why we have to perform this study, because this study or this extractable and leachable is having a potential impact on the drug product safety, efficacy, and it also impacts the regulatory compliance. So that's why it's very important to evaluate. Okay. So generally, as per the practice, first we are evaluating the extractables, and out of these extractables, we are doing study for the leachables and to qualify the leachables. But in some cases, the leachables are out of the extractables. In that cases, we have to identify leachables separately from the extractables.
So now we understood that extractable, what is extractable and the leachables. They are coming from the other sources than the pharmaceutical product. So can you elaborate more on what kind of sources they are coming from?
So we have already discussed about the container closure system, there are many give us the extractable and leachable. So first and most important is packaging components. For example, rubber stopper, plastic bottle, glass bottle, which are used for the packaging of a drug product. The second one is the container closure system. Even processing equipment can also contribute to give us extractable and leachables. And delivery device like inhaler, syringe, they can be a like potential source for the extractable and leachable. What is container closure system? So container closure system that directly contact the drug product either during the storage or during the administration of the drug product. So as they are directly contact to the drug product, that's why it is very important to evaluate this container closure system for the extractable and leachable study. See, we have two kind of container closure system. One is the primary component and second is the secondary component. So first we will see what is the primary component. The component that are directly contact to the drug product either during the storage or during the administration of the drug product. So they are directly in contact with the drug product. Whereas the secondary component, they do not directly contact with the drug, but do have the potential to interact with the primary CCS. So here the secondary component that contact with the primary component, and then primary component that directly contact with the drug and give us the probable extractable and leachables. Few example of primary CCS and secondary CCS to understand them better. So all the components in the meter, dose inhaler, the barrel, plunger, and tip cap in pre-filled syringes, the film and the pods on the IV bag, the aluminum tube and cap of the topical cream, the web and foil of the blister packet, they are all the example of the primary container closure and they are directly contact with the drug products. Whereas the printed labels, boxes, aluminum pouches, skids and overwrap during the storage of shipment and environmental exposure, they are the example of the secondary container closure. So here we can easily identify how the primary and secondary container closure they can contribute to the extractable and leachable.
Now we understood this. So these are the source extractable and the leachables come from the primary container closure and the secondary container closure. So this is very clear. So I just want to understand whether this all is a part of the regulatory requirement. Is there any specific regulatory expectation that along with the pharmaceutical product, we also evaluate, we have to evaluate the packaging material for the extractable and leachables?
Definitely, the regulatory have the guidelines to perform the extractable and leachable. So, if we discuss about the FDA guidance, FDA's health container closure system guidance like ICH Q3E and PQRI recommendation to design the SET and AET values. And USP has also defined the chapters such as 661.1 for the plastic material of the construction, 661.2 for the plastic packaging system, 1663 for the evaluation of the extractables, and 1664 for the evaluation of the leachables. So they can give us guidance how to perform these studies. And for the EMEA, WHO, and MHRA, they emphasize risk-based approach and patient safety for the evaluation of the extractable and leachables.
Regulatory has provided their expectation and the guidance for the extractable and the leachable. So now we want to discuss more about how these studies would be performed. So we will be discussing on the study conduct, what are the requirement of performing the extractable and the leachable studies.
Strategy, we cannot identify the potential extractable and leachable. And how to define then proper strategy? So there are two steps which are very important before performing any extractable and leachable study. So first is we have to evaluate the risk based on the available supporting data. So for example, any CCS, we have the COA, that material they have used for the construction of this CCS. So out of this material, first we have to identify is there any potential toxic impurity is there, what is its limit. That is the first strategy. And the second is after evaluating the COA of the materials, we have to design a strategy for the selection of solvents and method for the extraction. So based on the polarity of that particular CCS, we can choose a different solvent with a different polarity, and we can also sometimes use the buffers and pH to identify potential extractables. This is a generic study workflow to perform the extractable and leachable. So first is, as we have discussed, material selection and risk assessment based on the literature data available. Then second is extractable study. There are two kind of extractable studies: simulated and controlled extraction studies using solvents and temperature. The third part is after identification of the extractable, we have to evaluate leachables study, which is a real time and isolated conditions. Then analytical method development and validation of that method to identify the leachables. Then compound identification and its quantification. And the last part is toxicological risk evaluation after identification of any possible leachables. The first step is to define the study objective. So whether we have to perform this study in the controlled extraction condition or the stimulated extraction condition. So if we are going with the controlled extraction study, we have to identify and quantify potential extractables. Whereas for the stimulated extraction method, which mimics the real use condition to predict the actual leachables. Then selection of the material and the compound. So choose the materials that come into the direct or indirect contact with the drug product, as we have discussed about the primary and secondary CCS. Then prioritize the critical components such as primary containers, closures, delivery systems such as syringes, IV bag, and etc. Select the material and components. Generally, all the primary CCS components should be included in the extraction evaluation. Selection of secondary CCS can be used, but it depends upon the risk assessment evaluation. Then characterize the material. It is optional, but it is recommend to have all the possible extractables. So obtain material information, whether it is polymer type, adhesives, and processing aids, and perform initial chemical characterization. Example: FTIR, DSG, and TGA. Design the extraction condition. So it's defined based on the type of study. So for example, if we are performing the control extraction studies, then solvent selection should be of three range that should cover entire range of the polarity. So we can take a polar, for example, water, mid polar, for example, ethanol, and non-polar, example, hexane for the extraction. And the extraction conditions are typically extragated. So parameters that are used during the extraction, for example, temperature. Temperature should be between 40 to 70 degrees centigrade. Time generally 24 to 72 hours. Agitation should be a static, shaking, or stirring. And solvent ratio generally is 1 ml per centimeter square of surface area on per gram. As we have already discussed about the control, now we are going to discuss about the stimulated condition. So generally, we have to use actual drug formulation or placebo for the evaluation of stimulated extraction condition. We have to apply realistic use conditions such as time, shelf life, or duration of use of drug product, and temperature that are evaluated during the actual stability of the products, like for example, if it is a room temperature, a refrigerator, or any other conditions, that same conditions which was evaluated during the drug stability, that we have to taken in the simulated condition. Perform the extraction. So for that, first we have to cut, clean, and weigh the test articles, whether it is a rubber stopper or syringes, whatever it is. Then immerse it in the selected solvent under the control conditions, and the solvent, as we have discussed, has the same polarity of our testing material. Use brand controls solvents only to rule out the background interferences with our compound of interest. Filter and store extract properly. For example, if it is a light sensitive, then we have to use amber glass bottles or a light sensitive analyte. We have discussed simulated and controlled extraction condition. So what is the exactly difference between the simulated versus the controlled extraction condition? So simulated extraction condition and controlled extraction conditions are two different approaches to evaluating potential chemical compounds. Let's discuss about the controlled extraction condition in detail, and then we will discuss about the stimulated extraction conditions. So to identify the worst case extractables, the maximum amount and type of substance that could potentially migrate from a material under extracted conditions. The key characteristic of control extraction studies is use aggressive solvents, example hexane, ethanol, water, acid, base, and the conduct under extracted conditions which is having high temperature, long exposure, and agitation. Not intended to mimic real world uses but to maximize extraction of potential leachables. Often perform early in the product development to screen materials. Example using 100% ethanol at 70% 70 degree centigrade for 24 hours to extract from a polymer syringes. The simulated extraction studies. So generally simulate actual use of the condition of the product to assess what extractables may turn into the leachable under the realistic conditions. Key characteristic of the simulated extraction studies. So generally use product like conditions, actual formulation or placebo, relevant temperatures and durations, mimic real-time contact scenarios, storage durations or worst case use conditions, but still within the real world bounds. More relevant to the risk assessment for actual patient exposure. So example is exposing a drug container to a placebo situation at 25 degrees centigrade for 6 months to stimulate shelf-life conditions. A tabular format provided for the risk assessment. So risk associated with the route of administration versus risk of drug product interaction with the CCS. So when the risk associated with the route of administration is highest, then if we are using formulation like inhalation aerosol and solutions or injectable solutions and suspensions, then the risk for drug product interaction with CCA is very high. And when we are using sterile powders and powders for the injections or inhalation powders, the risk of the drug product interaction with the CCS is a medium. And if the risk associated with the route of administration is high, and we are using ophthalmic solutions and suspensions, transdermal ointments and patches, and nasal aerosols and sprays, then risk of the drug product interaction with the CCS is high. And if the risk associated with the route of administration is low, and if we are using topical solutions and suspension, the topical and inguinal aerosols and oral solution and suspension, then the risk of drug product interaction with the CCS is very high. For the topical powders and oral powders, the risk of drug product interaction with CCS is medium. And if we are using oral tablets and capsules, then risk associated with the drug product interaction with the CCS is very low. So
Great. This tabular form, the information is very useful to understand the risk associated with the route of administration, which is highest from the aerosol inhalation to the oral dosage form. So that gives better clarity. But I just want to understand: are both the control and the simulated extraction approaches required to evaluate?
is required to be evaluated because when we are using controlled extraction studies, it helps us to understand material risk and guide what to look for. While using simulated extraction helps us to understand patient exposure and support regulatory feelings by identifying a leachable available in the container closure system. To the solvent selection. The solvent selections are selected based upon the drug product with at least one of the solvent representing a worst case scenario. Because why we are using worst case scenario? To identify all the possible extractable present in the container closure system. The extraction conditions used are aggressive, typically reflux or oven incubation. So, what will be the end result? The end result is all the potential leachables will be identified. The PQRI guideline, the solvent selection should be based on the type of formulations. So, if the product is an organic propellant, then solvent selection should be a non-polar side, hexane, methylene chloride, and 2-propanol. If the product is an aqueous parenteral, then we have to choose the solvent which is more on the polar side. So, we can take a water or the combination of water with the alcohol, and we can also use some pH conditions to extract the as possible as extractables we can. And the guideline for the reasonable worst case. So if we choose a hexane for an aqueous product, then it is not a reasonable. And water for a water oil emulsion formulation, it is also not considered as a worst case. So in general, for the parental drug products, the solvent selection should be on the polar side, whereas for the orally inhaled or the nasal drug products, the solvent selection should be a non-polar side. So, examples for the extraction solvents for an aqueous drug product. So, if we are choosing a polar side, then water can be used 50 millimolar buffer at pH of drug product or 0.9% saline solution used as a polar solvent. And if we are taking intermediately polar solvent, then ethanol or the mixture of water, propanol, isopropanol in same proportion. And if we are choosing for the non-polar side, then hexane methylene chloride is the best option for the selection of a non-polar solvent for the extraction.
The control extraction and the simulated extraction we understood. So how can we identify the extractable and the leachables?
So now let's move on towards the identification of the extractables. So generally, we can use the mass spectral libraries such as NIST and Valley Library. We can perform the interpretation of the mass fragmentation patterns, isotopic patterns for the halogens and metals for the identification of the metal impurities, and also consideration of the polymer additives, lubricants, and antioxidants can also help to identification of the extractables. The tree for the identification from the uh, perform in the drug product matrix first, then more complex due to the interferences, require the sensitive, selective, and validated methods, and confirm identity with the standard if possible. So these are the steps to identify the leachables. Uh, Next is how to perform the analysis of the leachables in the drug product. So analysis of drug product stored in the CCS under the integrated storage conditions should be performed using the validated method. So here the difference is when we are performing the extractables, no validated method is required because it's just a starting of the study. But while performing the leachables, it is mandatory to perform the study using the validated method.
All right. So validated methods are specifically required for the leachables, not for the extractables, right? Because we need to quantify them and identify them. So can you tell us more about which kind of analytical methods are required to measure the leachables?
We know that like we have extractable and leachables, they are having different polarities, and we can also have the metals and evaporated impurities, non-evaporated impurities. So multiple instrumentation is required to identify all the possible extractables and then leachables. So starting with GCMS, gas chromatography, mass spectrometry, we can use for the volatile or semi-volatile compounds evolution. LCMS generally used for the non-volatile polar compounds. ICP-MS for the identification of the elemental impurities, FTIR, UV, and NMR for the structure elucidation, and finally HPS GCMS for the identification of the organic volatile impurities in the plastics. So, first of all, we require a proper laboratory setup for the evaluation of the extractable leachable. As we know that we require the different type of instrumentation and different techniques are involved. So, a typical GLP analytical laboratory having the capacity of the extraction of extractable leachable can suffice the purpose. And as we have already discussed about the instruments, LCMS, GCMS, IC, that is ion chromatography, and ICP-MS instruments are required to analyze sample after extraction to identify a possible extractable, leachable, and to detect them. We have discussed the sample extracts are analyzed by at least GCMS, LCMS, and ICP-MS in full scan analysis while we are detecting the extractables, and for the leachables we have to use MRM mode or other mode that can be quantified the amount of leachables present in a drug product. So to identify as many as extractables as possible and to semi-quantitatively determine the level of each acceptable leachable, we require all these techniques. Since the methods are designed to detect unknowns, these methods cannot be validated. So the development and validation of analytical method for the leachables, our goal should be the analytical method is having sufficient sensitivity, so can we can achieve LLOQ at or below AAT value. Extensive sample preparation may be necessary to ensure the sufficient sensitivity. So sometimes we can use the evaporation method to have the better sensitivity after extractions. The analytical methods are then validated with the goal to meet the ICH acceptance criteria for the specifically identification of the leachables. So targeted analysis methods are developed to detect the specific compounds that exhibit extreme genotoxicity. So example of such compounds includes polyaromatic hydrocarbons and nitrosamines, 2-mercaptobenzothiazole, and antioxidants. And we all know that nitrosamines are nowadays in the picture, and for the oil products it is required to be identify a possible impurities of the nitrosamines. So leachables are the analysis of volatile organic impurities. So that is called as VOC. Semi-volatile organic impurities, SVOCs. Non-volatile organic impurities, elemental entities, and inorganic anions, and low molecular weight organic acids. So that's why, as we have already discussed, many different kind of instrumentations is required to identify all the possible impurities. So specific ready-to-use standards are also available to evaluate the volatile, non-volatile, semi-volatile organic compounds, elemental entities, and inorganic ions, and low molecular weight organic acids. Because for the leachables, it is required to identify the quantity of the uh, leachables presence. That's why our calibration curve and QC are required to exactly or accurately monitor or identify the amount of leachable present in a drug product.
All right. So we understood what are the extractable and the leachables, and how to identify what are the sources. So that will help us to measure the extractable and the leachables. So in our next webinar, we are going to discuss how the method will be developed, how it is validated, and how can we measure the leachables in the product. So that will help. I request all our audience to take part in this discussion. And if you have any query or comment, please feel free to reach out to us. Our expertise can definitely help you to meet your objective. And I welcome you all in our next webinar. Will be coming live shortly. So see you then.